Thanks, Sabrina. My name's Reese, and with Dr. Tom Stanton, we're going to be co-chairing this next session on regional AMR challenges and solutions. And so without taking too much of your time, I'd like to now introduce Tanzina, who's going to discuss the phenotypic and molecular insights into different resistance phenotypes in Pseudomonas. So Tanzina, when you're ready, can you please share your slides?
Hello, can you see my screen?
Yep, we can see them, so you're good to go.
Thank you for introducing me. Hello everyone, I am Tanzina, a PhD student from UNSW Sydney. Today I'm going to share some research findings related to antimicrobial resistant differences between ExoEU and ExoS Pseudomonas aeruginosa in microbial keratitis. Let's start with a brief introduction. Microbial keratitis is an infection of the cornea which covers the iris and pupil. This infection can be caused by a number of bacteria. However, Pseudomonas aeruginosa is one of the most frequently isolated ones and responsible for 7 to 55% of cases. Pseudomonas aeruginosa keratitis can lead to perforation of the cornea within 2 to 4 days with potential blindness if appropriate treatment is not initiated. The pathogenesis of the Pseudomonas aeruginosa keratitis is associated with two exotoxins known as ExoEU and ExoS, which are usually mutually exclusive. Based on the presence of these two genes, Pseudomonas aeruginosa can be classified into two major groups. The first one is cytotoxic group, which contain the ExoEU gene and can kill the host cell because of its phospholipase activity. On the other hand, invasive strain contain ExoS gene, and this strain can invade and multiply inside the host cell because of its GAP and ADPRT activity. Let's delve into more information from literature. Research indicates that ExoS strain are more common in non-contact lens related keratitis, while ExoEU strain are more common in contact lens related keratitis. In addition, ExoEU strain cause more severe keratitis than the ExoS strain. So what's the reason behind? Might be the ExoEU strain have different resistance pattern and resistance mechanisms against antibiotics. However, this hypothesis requires further investigation. To prove this hypothesis, few studies were conducted previously, but those studies are limited on phenotypic detection of antibiotic resistance and genetic study mainly focusing on acquired antibiotic resistant genes, not detecting the target SNP to compare ExoEU and ExoS gene. So the question is, where is the gap in current literature? There is paucity of information regarding phenotypic and genotypic antibiotic resistance to compare ExoEU and ExoS strain of Pseudomonas aeruginosa in microbial keratitis. Therefore, it is important to study the difference of resistant pattern of these two groups because it might give you insight to develop a new therapy for keratitis infection. So, the hypothesis of my study was resistance and resistance mechanism to antibiotics will vary between ExoEU and ExoS strain of Pseudomonas aeruginosa in microbial keratitis. Based on this hypothesis, the aims of my study were set to determine phenotypic antibiotic resistance of ExoEU and ExoS, and then to compare these two lineages in microbial keratitis, and after that, to study the whole genome sequence of ExoEU and ExoS strain to determine which acquired resistant genes and target SNPs are associated with higher resistance to different groups of antibiotics. To perform the phenotypic study, I used 132 ExoS strain and 55 ExoEU strains, which were collected from Australia and India. MIC was conducted in 96 web plates to determine the susceptibility of those isolates against ciprofloxacin, levofloxacin, gentamicin, and tobramycin, which are from two different groups known as fluoroquinolones and aminoglycosides. MIC result indicated that ExoEU strain showed higher resistance to the all four tested antibiotic than the ExoS. And the difference was almost double in ciprofloxacin and gentamicin and more than double in levofloxacin and tobramycin. Interesting, isn't it? So it is important that which genetic material are associated with this resistance. Before going there, I would like to share the mode of action and mechanism of resistance in Pseudomonas aeruginosa. Fluoroquinolone antibiotics inhibit the DNA gyrase and topoisomerase enzymes, which are involved in DNA replication and separation of DNA to the newly formed cell, which are important for cell differentiation and cell growth. And aminoglycoside antibiotic inhibits the protein synthesis by binding to the 16S ribosomal rRNA of the 30S ribosome. So to counteract this action of antibiotic, bacteria use different resistance mechanism like intrinsic and acquired resistance mechanism. In intrinsic mechanism, bacteria use different efflux pumps which expel out antibiotic from the bacterial cell. For example, MAX A, B, C, D, E, F, X, Y expel out fluoroquinolone antibiotics, while MAX XY expel out aminoglycoside antibiotics. Positive regulatory genes MAX T, PAR, RS, ARMZ causes the overexpression of these efflux pumps. On the other hand, negative regulatory genes such as null CD, MAX R, S, Z, NFXB causes the downregulation of this efflux pump. However, bacteria can introduce mutation in any of these genes to increase the antibiotic resistance. For example, mutation in negative regulatory gene result in loss of function mutation, which actually increases the expression of the efflux pump, thereby increasing the antibiotic resistance. Bacteria can also introduce mutation in different target genes like gyrase A, B, PAR C, and E, which reduces the binding affinity of the fluoroquinolones to the target sites. In addition, bacteria can acquire different resistant genes which can carry out different functions like modification of the target sites or antibiotic structure or protection of the target sites. You can see here that RMT and QNR-VC genes are involved in target site modification and protection of the target site. There are different genes that are associated with modification of the antibiotic structure. Now coming to the genetic study result. For genetic study, I did whole genome sequencing of 20 exo-U and 19 exo-Pseudomonas aeruginosa from 187 keratitis isolates. And I used those whole genome sequence data to find out acquired antibiotic resistant genes that are associated with fluoroquinolones and aminoglycosides by RaceFinder database. You can see here that most of the acquired resistant genes are more common in exo-U group. And these two highlighted acquired resistant genes which confer resistance to aminoglycoside had significantly higher frequency in the exo-U group. However, there was no significant difference between these two lineages in terms of fluoroquinolone acquired resistant genes. For detection of SNP, target genes efflux pump regulatory genes are mapped with reference strain PA01 using Genius Prime tools. And you can see here that mutation in gyrase A, PAR C, these are the target genes, negative regulatory genes null C, MAX S, MAX Z, and efflux pump gene MAX B, C, E, and Y are exclusively present in exo-U group. And in other cases, they had higher frequency in exo-U group, except these two mutations which were more common in exo-S group. Among these mutations, these highlighted mutations are novel. So whether this highlighted mutation has any effect on the protein function, I did analysis of this mutation by shift algorithm and found that these 4 mutations are functional. Then I tried to find out whether this mutation has any association with antibiotic resistance. I did these things with Spearman's row test and found that the mutation in target gene gyrase A and PAR C were significantly large association with ciprofloxacin and levofloxacin. And in all other cases, the mutation showed moderate to strong correlation with both of these antibiotic resistance. Except this mutation, which are more common in exo-S gene, they showed negative correlation, which indicates that they are associated with the susceptibility of the antibiotics. I also did the same type of analysis with aminoglycoside resistance genes, that means the genes that are associated with aminoglycoside resistance, and I found that mutation in MAX Y, MAX Z are exclusively present in exo-U group, and with a variation of these two mutations, all mutations had significantly higher prevalence in exo-U group, and these two mutations had higher prevalence in exo-S group. Correlation study indicated that except these two mutations, all mutations had significantly large correlation with gentamicin, and in terms of tobramycin, they showed moderate to strong correlation. In addition, these two acquired resistant genes also showed association with aminoglycoside resistance. As exo-U strain had higher number of mutations, then I tried to find out the answer why. For this, I analyzed the DNA mismatch repair system genes MUT-L, S, and UVRD, which are involved in correction of the mutation during DNA replication. I found that even here, the exo-U strain had higher number of mutations, and the difference was significant in terms of MUT-L and MUT-S. So it indicates that because of the defective DNA mismatch repair system, that's why might be the exo-U group had higher number of mutations. So in conclusions, it can be said that exo-EU strains are more resistant to the antibiotic might be because of having different acquired resistant genes, acquiring higher number of mutation as well as novel mutation in the target genes and more mutation in the DNA mismatch persistent genes, which are associated with fluoroquinolone and aminoglycoside resistance. So I would like to take a moment to express my heartfelt gratitude to my supervisor, Professor Mark and Fiona, for their continuous support and guidance. And I am grateful to my all colleagues and lab members for their camaraderie. Thank you everyone for having patience. I'm happy to answer any question.
Thank you very much, Tanzina. Just a reminder, if you have any questions, please put them in the chat, and then Tom and I can read them out. So whilst we're waiting, I guess I can maybe quickly start with a pretty naive question, just because I'm not too familiar with the phylogeny of Pseudomonas, particularly aeruginosa. So do you think that so is the presence or absence of XO, U, or S toxins, is that involved in the kind of in the sequence typing or the clonal typing of these different Pseudomonas strains? And because you've shown like a nice correlation of their, well, not correlation, but a nice association of the kind of different AMR phenotypes within those strains. Can you maybe make a comment about that?
Okay, actually these two genes are part of type 3 secretion system. In Pseudomonas aeruginosa, there are 1 to 6 type 6 secretion system, which secretes different toxins that are associated with pathogenesis. So these exo-EU and exo-S are secreted by the type 3 secretion system. And in previous study, it's been found that the exo-EU strain causes more severe infection. So it's not associated with any sequence type because it's genes that are present in the chromosome, and based on the presence of these two genes, Pseudomonas pathogenicity is actually associated.
Okay, cool. Thank you very much. Any other questions?
Tanzina, I have a quick question. So on slide 11, you showed a really nice bar plot looking at association between fluoroquinolone resistance and mutations. Yes, yeah, and sort of mutations we found in these resistance determinants. So most of these are associated with increased fluoroquinolone resistance. But why do you reckon those OPRJ mutations are associated with increased susceptibility?
I think that this mutation are more common in exo-S group, and as they showed negative correlation, might be it has causes some changes of the efflux pump genes. That's why they are not able to expel out of the antibiotics. So that's why might be they are associated with the susceptibility of the antibiotic instead of showing resistance to the antibiotics.
That's a great answer. Thank you very much.
Cool, thanks, Tenzin. I think for the interest of time, we'll move on. So next, I'd like to introduce Abhinaba, who's going to talk about his work discovering different bacteriocins in Klebsiella isolates. So whenever you're ready, Abhinaba, please
Thanks,
share your...
Reese. Yeah, thanks, Reese. Yes, I'd like to share my slides now. Can you see it?
Yep, you just got to chuck them in presenter mode and then you're all good.
Yeah. Just a second. Yep.
Awesome. Thanks, Abhinaba.
So can you see it now?
Yep, perfect.
Okay. So thank you so much for this opportunity to present my work here. So a little bit background. I just started with so I'm a little bit. I had experimental. My background is pretty much experimental. I just started with bioinformatics a few like 1.5 years ago, and what I'm doing in Kelly White's lab is trying to find out bacteriocins in Klebsiella pneumoniae species complex, which are clinical isolates that we have identified in patients from Alfred Hospital. So we already know the importance of AMR and that Klebsiella pneumoniae is an escape pathogen and causes a lot of infections in hospitals. We have also gone through a lot of nice talks today that showed how Klebsiella pneumoniae can be a very good vector of AMR genes. And some of you might know that Klebsiella pneumoniae is a part of a species complex which share a lot of similarities with each other, and most of them are able to infect human beings. But some in Klebsiella pneumoniae, in a lot of these species complex, they're also very, very diverse because as you can see, and we have talked about a lot of clonal groups or like sublineages in a lot of talks. So you can see that some of them will have shorter branches, which means they are very they resemble each other closely than the rest of the population. So these clonal groups are very much varied in their antimicrobial resistance and everything. So it is important to know how can we develop antimicrobials against different clonal groups given our knowledge and genomic knowledge on all these isolates. Now, bacteriocins could be used as very potent antimicrobials to target Klebsiella. Bacteriocins are small toxic proteins that give the producing strain a competitive advantage over the other strains that are present in the same niche by producing this small toxic protein. And it is usually co-expressed with an immunity protein to protect its own self. So you can look at the diagram that how the immunity protein actually makes the bacteriocin ineffective by just blocking its active site. And thus bacteriocins have a specific activity spectrum. And if it does not have a cognate immunity protein, then it can target the bacterium that does not have a cognate immunity protein and basically kill it. So this has been highlighted in many experiments, but I'm going to show you this experiment done by M.N.T. Lee et al. In Hiroshima University that showed these are the different Klebsiella strains on each row, and they're affected by this bacteriocin producing strains on each column. And the heat map represents how resistant or sensitive they are against this bacteriocin producing strain. And as you can see that there is a variation in the effect of the bacteriocin resistant strain, as in they do not affect all the strains equally. So would it be cool that if only from the genomic data that we have, we can predict this kind of activity spectra from the isolates that we get from the hospital. This is what I've been currently working on. So employing a top-down computational genomics approach to identify these bacteriocins and the immunity proteins and predict the activity spectra of these bacteriocins in Klebsiella pneumoniae. So these are my broad aims, basically identifying the putative bacteriocin and also identifying it with a gene context. Identify the immunity proteins that is carried on with the bacteriocin, and then predict the bacteriocin immunity protein interactions and predict the activity spectra. So the methods are basically first looking for the bacteriocin. The way we did it is collecting the bacteriocin sequence database and using a remote homology detecting tool called hidden Markov model to look for to actually hunt bacteriocins in the clinical isolates that we have gathered from Alfred Hospital. We had like 452 genomes, and we use this HMM-based model to hunt the bacteriocins. And then we use a non-specific filter recommended by people who build the tool to filter out the false positives. And then we also applied a gene context-based filtering to filter out only the bacteriocins that contains an immunity protein or a secretion pathway. And now currently we are trying to look at the amino acid sequence variation in the bacteriocin, trying to see how similar or dissimilar they are in for each bacteriocin, looking at the gene location, whether they're on plasmid or chromosome, and finding the immunity protein that are associated with them. So this is basically an overall description of how many bacteriocins were found in the population. As you can see, most of the population, 63%, did not have any bacteriocin, which actually is pretty much in line with the experimental finding, which I think I forgot to mention, that they found bacteriocins in 32% of their isolates, where we found bacteriocin in about 37% of the isolates that we had. And as you see, the number of bacteriocins in a particular genome gradually decreases as like number of percent of population carrying a higher number of bacteriocin is low. There were 9 putative bacteriocins that we found on our hits, so now in our data set, and these are basically the description of the bacteriocin template that we have used. Mostly, we uncovered cloacins, microcins, and pyocins in our data set. Each allele, okay, so then we looked at the unique amino acid sequences. So for each bacteriocin group that we found, we grouped it according to unique amino acid sequences or alleles. And we see that each bacteriocin group has varies in the number of distinct amino acid sequences. And highest would be for these two groups, PB0577B and 538B, both of which are pyocin. Looks like pyocin kind of bacteriocins. Then we looked at how the coverage of, so how many sublineages in Klebsiella pneumoniae does each of the bacteriocins alleles occur in. So we grouped them by, so we bin them by the number of sublineages. And this on the y-axis shows how many different unique alleles that you will find. So for most of the groups, you see that you'll find highest number of alleles in only one sublineage. And that is indicative of the fact that these alleles are very infrequently shared across the sublineages. So yeah. So within each bacteriocin group, I tried to look at the diversity of alleles, as in how closely they resemble each other. So this is basically a heat map showing a darker color for if they are 95 to 100% similar, which means almost identical, would be the deepest blue, and less than 50% would be white. So as you can see, for this particular group, we have a high variety of alleles in that group. Whereas in a different group, we see most of these alleles resemble each other. So the comment on here would be each bacteriocin group seems to be different in their variations of alleles. Now looking at, so this is again the same tree that I showed before. So it's looking at the different sublineages of Klebsiella pneumoniae that we got from Alfred Hospital. And this is mapping all the different clusters of amino acid groups, all the clusters that are 80% or more sequence similarity within each bacteriocin group. And you can see that there is quite some amount of clustering closer to each sublineage. So as you can see over here, that all these similar looking bacteriocins are very close in one sublineage. Although, and you can also see that that this is a significantly less number of unique bacteriocin clusters spread across the whole population, which is a good thing because that means that we have highly specific amino acid variation in our subpopulations, which makes it which would probably make it easier to find a good bacteriocin candidate that would work against the other population. So we also looked at whether this bacteriocin is present in a plasmid or a chromosome, because if it is present on a chromosome, it would be a better candidate as it would be less labile and less would have a less tendency to spread in a population. So we looked at that. We see quite a number of bacteriocins are present in plasmid, where some is present in chromosome. But the most interesting thing was none of this bacteriocin group had both chromosome or plasmid containing bacteriocins in them. We also looked at the number of bacteriocins for each isolate. As you already have seen in a more less detailed picture before. You see this group, particularly this group of sublinases carrying at least 4 bacteriocins, but most of them carry just 1 or none. So, okay, so going back to summary, we saw that we have a 37% of bacteriocin that we found among the clinical isolates in our database. Each bacteriocin group was spread across only a minority of isolates. Bacteriocins often exhibit a high diversity of unique amino acid sequences or alleles. They are not frequently shared across sublineages that indicate a strain specificity, which would be important for targeting other KPSCs. Future directions, we are going to determine the number and diversity of immunity proteins that is co-located with these bacteriocins. And based on that, we want to generate a predictive framework of the bacteriocin activity spectra across this population. And finally, maybe later on, we would do an in vitro study to confirm if these bacteriocins are actually active and if our model makes sense. So with that, I would like to thank the group, Dr. Kelly Wires, Dr. Ben Vizina, who are my supervisors, as well as Tom, who is here currently as a chair, and the rest of the group, Monash University for the grant, and all the rest of the helpline for the research. Thanks a lot, and I would like to take any questions.
Thanks, Abhinava. I just have one quick question because we're just in the conscious of time. So with your different alleles that you've identified, are you then planning to look at where those changes are in terms of the structure of the different bacteriocins? So do they map to regions that would recognize the receptors on the different target hosts or more functional domains? Can you maybe make a comment on that?
Yes, great question, Reese. We will actually look at that. What I have not stated for the interest of time was we are planning to predict the structures for each bacteriocin clusters. And then we are trying to figure out the most common part where these bacteriocins might differ for within each bacteriocin group where the alleles might differ. So we're trying to figure out the hotspots. Which is a little bit future part of this project. But yeah, that's in the plan.
Cool. Thanks, Abhinava. I think in the interest of time, we'll move on.
Cheers, Reese. Yeah, thanks everyone so far. I'm gonna be introducing our last 3 speakers of this session. And just as a gentle reminder, please try and keep your talks to 10 minutes so we can have some discussion afterwards. So we're going to be moving on to Abby Mole, a PhD student from the University of New South Wales. I think I've got that correct. Who's going to be talking about molecular profiling of antimicrobial resistance and virulence factors in Pseudomonas aeruginosa isolates from keratitis patients. Away you go, Abby. Thank you.
Okay, thank you. Is my slide visible for you?
Yep, looks great.
Okay, thank you. Good afternoon, everyone. I'm Abie. Today I'm going to share our recent works, molecular profiling of antimicrobial resistance and virulence factors in Pseudomonas aeruginosa isolated from keratitis patients. As we know, this opportunistic pathogen is a gram-negative bacteria, which is well known by its swarming motility and biofilm formation. And the report shows that this opportunistic pathogen uses various virulence strategies, such as type 3 secretion system, the iron uptake system, the type 4 pili, and the protease, flagella, and other proteins to establish infection on different bodies of the human being. This opportunistic pathogen causes a wide range of infections, including keratitis. Keratitis is an inflammation of the cornea, which leads to corneal scarring and perforation. If it left untreated, it leads to visual impairment and then blindness. Surprisingly, a recent outbreak in the United States resulted in 4 patients' deaths, 4 patients' eyes to be removed, and 14 patients' loss of vision. Recently, multi-drug resistant Pseudomonas aeruginosa isolates has been increased alarmingly. Interestingly, multi-drug resistance and XDR Pseudomonas aeruginosa strains detected in ocular isolators. Specifically, the aminoglycoside resistance genes, beta-lactam resistance genes, chloramphenicol resistance genes, ciprofloxacin resistance genes were detected in ocular Pseudomonas aeruginosa isolates. So the question is why this Pseudomonas aeruginosa becomes MDR and XDR. There are 2 possibilities. One possibility is it is intrinsically resistant to several antimicrobial agents, and also this opportunity pathogen is highly adaptable and can acquire resistance via mutation and horizontal gene transfer mechanisms through conjugation, transduction, and transformation. So the growing incidence of multi-drug resistance Pseudomonas aeruginosa is recently a public issue, and this results in treatment failure and profound consequences. So it needs an urgent development of new antibiotics and urgent solution to control the circulation of these multi-drug resistance strains. Despite the phenotype characterization has been done before, the genetic basis of antimicrobial resistance and violence factors remains uncertain and not fully explored. Therefore, we are currently working to unravel the novel violence factors and resistance mechanisms in ocular Pseudomonas aeruginosa isolates. So to do this, we include 70 whole genome sequencing data sets of Pseudomonas aeruginosa obtained from corneal samples, and then we use bioinformatic analysis for different, like for annotation proca, for AMR gene profile card, and other pipelines for virus factor characterization, hospital horizontally acquired antimicrobial resistance genes, as well as for pangenome analysis. So the pangenome analysis shows that the ocular Pseudomonas aeruginosa isolates contain high number of cloud genes, which reflects the genetic diversity and evolutionary flexibility within Pseudomonas aeruginosa strains. Moreover, multilocus sequence typing analysis shows that most of the isolates belong to distinct sequence types. However, some isolates represented new sequence types, which indicates that still there are uncharacterized genetic diversity within the Pseudomonas aeruginosa population. To evaluate the evolutionary relationship between those ocular strains, a phylogenetic tree was constructed, and we found distinct clusters, 3 distinct clusters, and clades and branch lengths, which reflects the evolutionary divergence between those ocular Pseudomonas aeruginosa strains, as well as strains from similar multilocus sequence typing. They are clustered closely, which suggests they are genetically similar or they share a common ancestor. And also long branches in some strains we observed, which may be due to more genetic changes within those strains, and also short branches also observed in some strains, which may share similar evolutionary relationships or more close to each other. The antimicrobial resistance gene profile analysis shows that the ocular Pseudomonas aeruginosa isolates contain several antimicrobial resistance genes against different class of antibiotic. For example, we detect different antibiotic resistance genes against beta-lactam antibiotics, aminoglycoside antibiotics, fluorouracilone antibiotics, peptide antibiotics, as well as for disinfectant and antiseptic resistance genes. Surprisingly, the majority of the identified antibiotic resistant genes in ocular Pseudomonas aeruginosa isolates involved in antibiotic efflux pump systems. The red color in this heat map shows that the abundance of the resistance mechanisms in each strain. And also, we identified the acquired antimicrobial resistant genes in Pseudomonas aeruginosa isolates using a RAS-finder, and we found some beta-lactam resistant genes like BLPO and BLOXA gene variants, as well as different resistant genes for aminoglycosides, fluoracil, chloramphenicol, and phosphomycin. So the question is where those acquired antibiotic resistance genes originally get by these ocular strains. So we just try to identify some of the horizontally acquired antibiotic resistant genes to identify the source. So we use a BLAST as alignment, and then most of the genes that identified in the as acquired antibiotic resistant genes, they are related with other Pseudomonas aeruginosa strains. So possibly these antimicrobial resistant genes may be acquired from other Pseudomonas aeruginosa strains through horizontal gene transfer mechanisms. And also we detect a considerable number of mobile genetic elements among ocular Pseudomonas aeruginosa isolators, specifically the insertion sequences and the transposons are common in the ocular Pseudomonas aeruginosa strains. Those mobile genetic elements can move or jump within and between genomes, and they are playing a key role in bacterial evolution and the spread of antimicrobial resistant genes, and often they are associated with horizontal gene transfer mechanisms. Therefore, those mobile genetic elements may contribute to the emergence of multi-drug resistant strains by disrupting the genes or regulatory sequences when they are inserted, or through cut and paste mechanisms. They contribute for genetic variability and emergence of resistant strains in Pseudomonas aeruginosa. And lastly, the valence factor analysis shows that the majority of the valence factors in ocular Pseudomonas aeruginosa isolates are flagellary proteins, type 3 secretion system, the iron uptake system, the type 4 ply, and other valence factors were detected, and those valence factors may help this opportunity pathogen for the successful pathogenesis or colonization of the human body, and leads to infection in the cornea and other parts of the human body. In conclusion, the inherited and the acquired antimicrobial resistant genes, along with several structural and secretory valence factors, indicate the ability of the ocular Pseudomonas aeruginosa to emerge as multi-drug resistant infectious strains. Therefore, future experiments are better to focus for the development of new antibiotics targeting new mechanisms, specifically for the identified mechanisms and the identified valence factors targeting the efflux pump systems, as well as some valence factors like type 3 secretion system. So those may help to tackle the spread of these antibiotic resistant genes, antibiotic resistance in Pseudomonas aeruginosa. Furthermore, long-read sequencing like nanopore sequencing, PacBio sequencing may help to induce characterization of these opportunistic pathogen for its associated with valence factor and antibiotic resistance to tackle the rising incidence of multi-drug resistance Pseudomonas aeruginosa infections. And finally, I would like to thank my supervisor, Professor Fiona and Professor Mark, as well as School of Optometry and Vision Science, University of New South Wales, the organizing committee for the symposium, and also the IPFL program and other stakeholders. Thank you so much.
Thank you, Abby. That was a great talk. Do we have any questions from the audience or in the chat? No, okay. I've got a quick question. So we've heard a little bit about IS elements already in E. coli. So it's really cool that you pointed those out as well. With the IS elements that you found in your gene, Pseudomonas aeruginosa genomes, was there any evidence you found of those disrupting any genes that were important for virulence or AMR, or perhaps in the promoter regions, driving the expression of any genes important for those phenotypes?
Yeah, we are currently at early stage for characterization. So as we know, the mobile genetic elements can carry different antibiotic resistant genes. Maybe the insertion sequence transposon and plasmids may carry different antibiotic resistant genes, but specifically for the specific promoter or the specific antibiotic resistant genes carried by those mobile genetic elements has not yet been identified, and we will focus on future.
Oh, that's great. Yeah, no, they're quite difficult to characterize sometimes.
Yeah.
So To has put a question in the chat. To has asked, you mentioned long read sequencing in future work. What is the purpose of doing long read sequencing for this project?
Yeah, currently, we are working for the genotype characterization, specifically targeting for antibiotic resistance and some violence factors. So just most of the current sequencing data included is done by Illumina short-read sequencing. So if we use nanopods like long read like PacBio, it covers more large genomes of, or we can sequence the whole genome sequence of the bacteria. So it gives more better information for characterization. So for future, just we'll try some strains uh, for long read sequencing.
Also might help you figure out what those IS elements are doing as well. All right, thank you, Abhi. In the interest of time, we'll move on to Shyam Mishra, a PhD student from the University of New South Wales, who's going to be talking about antimicrobial sorry, I don't really mispronounce that, against drug-resistant yeasts of medical importance. I probably should have looked that word up before saying it. All right, thank you, Shyam. If you want to share your screen.
Hi, yeah.
Maybe you can help me pronounce that.
I think this is the one. Is it same?
Yeah, it looks great. Thank you.
Okay. Good afternoon. Today I'm talking on antimicrobial peptidomimetic against drug-resistant yeast of medical importance. So as we know, these days, like besides bacteria, like different fungi, particularly yeast cells, they are also emerging. For example, Candida auris, like it's just been around 15 years that it has made a huge impact in clinical treatment, and also the well-known Candida albicans is there. And again, all the time, like taxonomically, very non-static, some species of Candida, for example, glabrata or Candida krusei, as well as Tropicalis, Parapsilosis, Guillermondi. So there are different Candida species which are causing infections, and they are being listed by WHO as critical priority pathogens. Because many of these strains are resistant to different antimicrobials, which are commonly used, for example, casofungin or amphotericin B or fluconazole. Therefore, like scientists are trying some alternatives, and in that round, so we are also looking for different peptide mimics. These peptide mimics are in fact related to antimicrobial peptides, but they differ from the peptides in that these are resistant to the different proteases that are present in the body fluid, so that they provide very much biostability when they are used in vivo. So for this study, like I used two different peptidomimetics. One was TM8 and another RK758. These, as we can see here, okay, this TM8 consists of long chain of alkyl chain, which simply help to bind with the cell membrane, as well as RK758. It is a brominated compound which is very much effective with respect to which was found to be very much effective with respect to virus as well as bacteria. Therefore, we try to see how effective these compounds are with respect to fungi. So for this, like different species of Candida were chosen, and they were resistant to, as we can see, fluconazole or amphotericin B or casofungin with respect to different species. So if we focus with respect to fluconazole, so our compound was showing very good activity. So the MIC, geometric mean MIC with both of these compounds, TM8 and RK758, was around lower than 10.0 microgram per mil. If we see like Pichia or like Candida krusei, to which fluconazole is intrinsically resistant, having MIC of 128, our compound is showing good activity, as well as for Candida auris, below 10 microgram per mil. So this subjected us like this, simply made us very enthusiastic to look how these compounds will act as synergistic, whether this compound will act as a synergistic compound with respect to the conventional antifungals. For that, we tested these compounds, both TM8 and RK758, with casofungin and amphotericin B as well as fluconazole, and we found that these compounds have got very good activity. Like, for example, TM8 showed good synergy with amphotericin B, and RK758 showed very good synergy with fluconazole. In almost all the cases that we tested, they showed either synergy or partial synergy. And the best part was there was no antagonism in any of these combinations against any of these strains. To further confirm this combination outcome, we further did another method, like we did time kill assay with the use of one particular strain of Candida using fluconazole with TM8 or RK758, and we found that at the synergistic concentration, the microbial load or the Candida load was decreased from 10 raised to the power 5 to 10 raised to the power 2, around 2. So with the 3 log reduction. However, at the same sodium-MIC concentration, when the compounds were used in singles alone, so they could not inhibit the microbial growth with the rise in the number of Candida. So this simply means that we found a reproducible result with another method, time kill assay as well. That means these compounds are really acting as synergistic agent. Now, to identify what exactly was the mechanism of these compounds, we did sorbitol protection and ergosterol binding assay. Sorbitol protection is basically done to see whether it is acting on the cell wall, and ergosterol binding assay is done to see whether it is acting on the cell membrane. So when the MIC was done in the presence of sorbitol, there was no change in MIC up to 7 days. That means sorbitol is not acting as osmoprotectant. That means it is probably the compound is not acting on the cell wall. However, when ergosterol was placed exogenously, then we found that the MIC increased by 4-fold in all the cases, which means the compound is binding with exogenous ergosterol, showing its affinity to ergosterol. And ergosterol is present in the cell membrane of fungi. Here we can see that means both of these compounds have got the affinity to ergosterol, bind them, then they cause membrane permeabilization and dysfunction, and finally lysis of the cell. As both of these compounds show the similar mechanism action, we further took 758 for our further studies. And to confirm their mode of action with respect to cell membrane, we did flow cytometry analysis with the use of live-read stain, cyto9 and propylium iodide, in which we found that in due course of time and also with higher concentration of the compound, so it is having, it is showing good killing of the cells with the reduction in number of viable cells and increment in the number of dead cells. So flow cytometry supported that it is acting on the cell membrane again. To further confirm and to further see what other anatomical and morphological changes can be seen after the treatment of 758, we did transmission electron microscopy. After 2 hours incubation or exposure of this compound with Candida auris, we found that with respect to control cell, so the cytoplasmic clearing was seen, and also like in many of those images, we found that there was some intracellular, like in the inner membrane, there were some membrane blebs showing that there is some kind of stress happening there. And also in the nucleus, there were like some pores were seen, and also irregularities in the inner membrane of the cell membrane. Further, so many of the times, like these cytoplasmic content were found to be accumulated or flocculated at the periphery of the cell membrane, showing some kind of stress that is going through in the Candida after exposure of the compound. Besides that, like the best part of these antimicrobial peptides is they do not have single mechanism of action. They might not simply act on the membrane, but they might be having multiple modes of action. As for example, here we can, as I already said, that in different cellular organelles, so they are showing activity. So even if any of the cellular organelle or any of the gene which code for some mechanism, they become resistant, another mechanism is still there of this compound, which will confer killing of this microorganism. To further check if this agent, Candida, can develop resistance to these compounds, so we did sub-passes in the presence of sub-MIC concentration of all these compounds for 30 days, and so that the Candida auris, it was very hard for it to develop resistance. It only showed the increment of MIC by 2 times. However, other conventional antifungals, they showed increment in the MIC by 4 to 8 times. So this is again a good finding. Now for the, like, if these agents are effective not just against planktonic cells, but also against biofilm formation or to prevent the attachment of candida against any surface. So we did anti-adherence, premature biofilm disruption, and mature biofilm disruption assay. So I would like to focus here. So here we can see that more or less at the similar concentration, ranging from 0.5 to 8 sometimes, but more or less 0.5 to 2 times the concentration of MIC that was effective against planktonic cells. These compounds were very much effective to eradicate mature biofilms. That means they are not just effective against planktonic cells, but they are also acting as anti-biofilm peptidomimetic, which is again a great finding. Because in many other times, as we know that for antifungals or antibiotic, for biofilm to disrupt the biofilm, it might take around 100 to several thousand concentration of MIC of the compound. So here we are getting at a very low concentration. That means it is effective even against biofilm. Further, to explore further mechanistic activity, we treated this Candida auris with our compound and did the transcriptomic analysis. And in the transcriptomic analysis, we found a total of 331 differentially expressed genes. They were identified with 195 upregulated and 136 downregulated genes, indicating that the compound significantly affected the gene transcription of Candida auris. So additionally, geoterms and KGG pathways analysis were also used to identify the function of like differentially expressed genes. And what we found basically was, so the metabolic and catabolic genes like processes, they were basically downregulated, whereas the RNA processing phenomenon, so those genes which code for RNA processing, they were upregulated. That can signify that there is some kind of stress that is going on in the Candida after the exposure of this compound, trying to protect themselves from different, from this particular factor. So despite one of the regions of their metabolic, metabolic lagging in the metabolic pathways, so that can simply make the Candida to go to the biofilm formation or the tolerance phase or persister phase. However, as we already, I already saw that they were effective against biofilm as well. So even the fungi tried to develop some resistance, so they were not able to do so. So further, like I would like to add one more thing here, like since these compounds were found to be effective against cell membrane, so that means it might have some activity related to amphotericin B. I still have to compare how the transcriptomic differences is there between its activity with respect to amphotericin B and this compound. That process, like that study is still going on. But to conclude, so the two new compounds, RK758 and TM8, they were effective against both almost all Candida species that I tested, against both planktonic as well as biofilm cells. They act on the fungal cytoplasmic membrane with cellular disruption, and not just that, they have got multiple modes of action. Even intracellular mode of action is also there. And it was very hard for the fungi to become resistant to the compound. The peptidomimetic was efficient in preventing biofilm formation, even attachment, also the disruption of premature or biofilm formation or disruption at a very lower concentration. And thank you so much. This is our team of Professor Mark Wilcox. Thank you. I would like to thank everyone. Thank you. Any question, please?
Thank you, Shyam. That was a great talk. I really like this orbital ergosterol experiment. To has asked another question in the comments, and I think it's something that we're all wondering. What could any potential resistance mechanisms to these for Candida to become resistant to these novel compounds? What do you think they could be?
What could be the possible resistance mechanism you mean to say?
Yeah.
Like it has got any, normally what we say that for antimicrobial peptides or antimicrobial peptidomimetic, again, like it is very hard for any bacteria or fungi to develop resistance. But again, it is not absolute. With due course of time, definitely, like in other bacteria or fungi, so those mechanisms, what are there, that is there. Like with the development, like the Candida or the fungi can go to the persister form or tolerant form, so that can be there. Even the ergosterol, there can be some changes with the enzyme, as we can see for fluconazole, like lanesterol dimethylase enzyme that is there, the change in the mutation that is conferred resistance to fluconazole. So that kind of thing might happen. We never know. But till now, what I can say that this compound is not showing, Candida is not showing resistance to these this compound. However, still we have to work, like we have to see that still we cannot at this point say that it will not develop resistance, but that is always a spontaneous mutation. Such kind of thing can always happen with the all known mechanisms that we know against fungi or anything. Yeah.
Fantastic, thank you. I do just have a very quick question. Why do you think the combination of TM8, which was I think your first compound, with fluconazole, why do you think that was so synergistic?
Yeah. So for fluconazole basically act on the cell membrane, particularly ergosterol. And as we have, as I have already shown that again, the TM8 as well as RK75, both are acting on the cell membrane. So that is there. But again, this compound with respect to transcriptomics, like again, I have not shown the full data, but with respect to transcriptomic as well as with respect to the transmission electron microscopy, we saw different changes in the intracellular details, particularly with transmission electron microscopy. So that simply can help us to make a conclusion that it is not just acting on the cell membrane, but different other components of in the cell. That is, it is not just acting on the membrane, but it's also acting on the intracytoplasmic component. That is so that even if fluconazole or amphotericin B or any compound that is not active, so these compounds can bypass that pathway, and also they can lyse the cell membrane and also act in the intracellular things. So that is the, and coming again to your question, they have got very good like alkyl chain, like decamer is there, like long fatty chain. So that they have got very good affinity to bind with the cell membrane and to lyse the membrane, to have binding and then lysing. So probably that has resulted in that synergy. Yeah.
That's great. Lots of hope for the future. Okay, so next up we have Winnie Omari, a master's student from Tanzania, who's going to be talking about combating antimicrobial resistance in the Katavi region, challenges and strategic solutions. So Winnie, if you'd like to share your screen, that'd be great. Brilliant, thank you.
Hello. Okay. Do you hear me?
Yeah, we can hear you fine, thank you.
Yeah, okay, thank you. Thanks for this opportunity, and I'm going to have some discussion with you on the challenges and the strategic solution on the AMR in the region of Katavi in Tanzania. Sorry. Having some difficulty here. So just a few minutes. I can't move my slides. Yeah, thank you. So I think we're all aware of the antimicrobial resistance. It is a global threat, and we have a lot of morbidity and mortality cases. So Katavi regions, we face a lot of challenges, including limited resources on tackling the antimicrobial resistance. So the objective of this study was just to assess the challenges of AMR, as well as to propose the solutions that will help us to improve the public health on the fighting against the AMR. And it was just a cross-sectional study in the Katavi Regional Referral Hospital, the western part of Tanzania. It was in 2023, and just laboratory analysis of the clinical sample from the patient. We identified the resistance patterns, and also we had surveys to the health workers on their understanding how they prescribe the medication to the patient, as well as we had some sort of interviews with the patients to know whether they have any awareness about the AMR. And we performed the antimicrobial sensitivity testing. We had the Kibibauer techniques. It was a manual, and we don't have, as I said before, good infrastructure facilities are not very supportive. So we performed it. Also, we had the factors that we analyzed in our findings, including the prescription practices from the physician, compliance from the patient, as well as the IPCs. We had the findings with high prevalence of ESBR, probably 48%, and the carbapenem resistance in Enterobacterales of about 16%. I didn't put it here, sorry for that. And also we had the MRSA and the inducible macrolide lincosamide septogram resistance. This was observed most on the Staphylococcus aureus, but also we had a very few number of the Coagulase negative staph species resistance to the the methicillin groups of penicillin. Also, the resistance rate was exceeding 60% for commonly used antibiotics in our region of Katavi. And also, as you can see, the challenges here of a prescription of antibiotics. This is because we have the standard treatment guideline at the national level, but most of the physician does not rely on it. They just prescribe the antibiotics. And also we found the overprescription of antibiotic of the patient themselves without attending the health centers. We captured that information through the interviews. And also lack of adherence to IPC. Of course, we had now, our hospital has about 3 years from it is opening, but it was this year we had the IPC team in our hospital. It was a difficulty. So public awareness about AMR is very low. This is a very big challenge in our community because most of the people does not seek for the prescription of this antimicrobial agents. They just take themselves and they treat themselves without asking prescription advice from the physicians. So what were the strategic solution? So just we need to enhance our antimicrobial stewardship program in Katavi region, starting from guidelines for appropriate antibiotic use, and also to train the healthcare workers. Another challenge we faced, it was the inability of the laboratory personnel, how to detect, to screen, and to confirm any resistance patterns from the antimicrobial sensitivity testing. And also, we need to implement the robust infection prevention and control. We need to establish and enforce the IPC protocol in healthcare setting. This now includes not my hospital only, just the whole region of Katavi, and the regularly monitoring and evaluation on the IPC measures. And for saying that, we had the previous training on the IPC measures among the healthcare workers in Katavi region. And also, we want to conduct community awareness campaigns on the proper use of antibiotics. At this part, we'll be facing the community, general community, just to have some conversation on the threat of AMR. And we'll be doing this through media as well as community outreach. So, AMR is still a significant problem in Katavi region because at least for every week we have 6 to 10 deaths, specifically the pediatric. So it is still a significant threat in our region and in Tanzania in general. But also the multi-drug resistance bacteria, there is high prevalence, of course, the carbapenem resistance in Enterobacterales as well as the MRSA. And also we need now to have a multifaceted approach, including collaboration between anyone who has that heart to think that AMR is a global problem. So anybody can lose life and so on. So it has to be a collaboration. And also what we expect from this approach is just effective implementation. Only this one can reduce. We want to reduce and improve the patient outcome because that is our work. And thanks so much. welcome for the question. And that's the end of my presentation. Thank you.
Thank you, Muni. That was a fantastic talk. Do we have any questions in the chat? I can't see any. Does anyone have any questions in the meeting? Whilst we're waiting, I'm just curious, for the Katavi region, in your opinion, what is the most effective way to engage with the local community on antimicrobial stewardship issues? Because this is the challenge, regardless of where you are, and obviously each place you have to engage with the community on this issue in a different way. So, in your opinion, how would you implement this?
Yeah, as you said, it is a global concern, and recently, I think the community outreach to me will be a better way, because I'm talking about my country. I don't know about the Eastern or Western countries, but most of the people don't even have a little understanding on the AMR. So the only way to reach to them is just to face them on the community. Of course, we are trying to go in the markets, because it's a place where we can find a lot of people. So going there and telling them AMR this, AMR that, it's sometimes we go to schools also, talking to the children at younger age to tell them the importance of not taking antibiotic without a prescription. I think community outreach to me is the best way.
That's really interesting. Thank you very much. Does anyone else have any questions? So Rain has asked a question in the chat. I think they are asking you to expand a little bit on your sampling methodology and your how you're differentiating between environmental and human samples.
Yes. Can you repeat it, please?
So could you describe your sampling methodology in a little bit more detail, please?
Yeah, okay, thank you. So we were taking the clinical samples for the patient that were admitted in the hospital for the whole year of the 2023. So whatever the clinical sample, regardless of the sample type, came to us and we found any pathogen, it was just our part of this scan. So we included every pathogen from every sample. And we performed just the routine laboratory activities. At the same time, we used it to go to the wards to ask the patient about their awareness on the AMR, if they had a previous use of antibiotic before coming to the hospital. And also we had the physician. So we used it to ask the physician directly how they tackle this infection. Most of them gave us a very good explanation, but some of them didn't have a good explanation, specifically on the standard guidelines of the nation on the treatment of any infections.
So you collected their, I presume you collected their past information about their past antibiotic use. Did you see a correlation between past antibiotic use prior to admission to the hospital and sort of rates of AMR infections?
Yeah, exactly. For instance, we had patients who were requested to perform the blood culture, but they came to the hospital with already had on the prescription, their own prescription, self-prescription of antibiotics. So we found the negative growth of microorganisms. But after a certain time, we collected the sample again and we found they are still sick because they used the wrong medication. So at the time we collect the sample, the second time we captured the microorganism.
Okay, that's great. Yeah, so you have evidence of incomplete sort of chemotherapeutic usage. Very interesting. Okay, so I think we will break until 4 p.m. So we're going to start again at 4 p.m. Sharp for the lightning talk session. Thank you very much, everyone who attended this session, and please give a big thanks to all of our fantastic speakers and my co-chair, Reese. Thank you very much, everyone.